We are back with glycolysis as a continuation of the topic. We just started it. We're just going to complete it now. The energy generation phase. Glyceraldehyde 3-phosphate dehydrogenase converts glyceraldehyde 3-phosphate to 1,3-biphosphoglycerate. This enzyme brings about two reactions, namely oxidation, where the hydrogens are transferred to NAD+, and phosphorylation of substrate, adding 1-phosphate moiety. The step is important as it is involved in the formation of NADH plus H plus and a high energy compound of 1,3-biphosphoglycerate. And this enzyme brings about two reactions, namely oxidation and where the hydrogens transfer to NADH, and the phosphorylation. Iodoacetate and arsenate inhibit the enzyme glyceraldehyde 3-phosphate dehydrogenase. In aerobic condition, NADH passes through the electron transport chain, and 6 ADP, 2 and 3 ADPs are synthesized by oxidative phosphorylation. And the enzyme phosphoglycerate kinase acts on 1,3-biphosphoglycerate, resulting in the synthesis of ATP and formation of 3-phosphoglycerate. This step is a good example of substrate-level phosphorylation, since ATP is synthesized from the substrate without the involvement of electron transport chain. Phosphoglycerate kinase reaction is reversible, a rare example among the kinase reactions. 3-phosphoglycerate is converted to 2-phosphoglycerate by a 3-phosphoglycerate mutase. This is an isomerization reaction involving transfer of phosphate group from position 3 to position 2 in the substrate. The enzyme requires Mg2+, that is magnesium. The high-energy compound phosphoenolpyruvate is generated from 2-phosphoglycerate by the enzyme enolase. This enzyme requires magnesium or manganese and is inhibited by fluoride. For blood glucose estimation in the laboratory, fluoride is added to the blood to prevent glycolysis by the cells so that blood glucose is correctly estimated. The enzyme pyruvate kinase catalyzes the transfer of high-energy phosphate from phosphoenolpyruvate to ADP, leading to the formation of ATP. This step is also a substrate-level phosphorylation. Pyruvate kinase requires K+ and either magnesium or manganese. This reaction is irreversible. Conversion of pyruvate to lactate significance. The fate of pyruvate produced in glycolysis depends on the presence or absence of oxygen in the cells. Under anaerobic conditions, lack of O2, pyruvate is reduced by NADH to lactate in presence of the enzyme lactate dehydrogenase. Competitive inhibitor oxamate. The NADH utilized in this step is obtained from the reaction catalyzed by glyceraldehyde 3-phosphate dehydrogenase. The formation of lactate allows the regeneration of NAD+, which can be reused by glyceraldehyde 3-phosphate dehydrogenase, so that glycolysis proceeds even in the absence of oxygen to supply ATP. That's pyruvate. The occurrence of uninterrupted glycolysis is very essential in skeletal muscle during laborious exercise, where oxygen supply is very limited. Glycolysis in the erythrocytes leads to lactate production. Since mitochondria, the centers for aerobic oxidation, are absent, brain and retina, skin, renal, and medulla and GIT tract derive most of the energy from glycolysis. Production of ATP in glycolysis. Under anaerobic conditions, 2 ATP are synthesized, while under aerobic conditions, 8 or 6 ATP are synthesized, depending on the shuttle pathway that operates. Glycolysis and shuttle pathways takes place in the cytosol. The NADH produced by the enzyme glyceraldehyde 3-phosphate dehydrogenase is utilized later by the enzyme lactate dehydrogenase, and NAD+ is regenerated so far as the synthesis and utilization of reducing equivalents are coupled. Glycolysis proceeds uninterrupted and the end product is lactate. The perfect coupling is possible only in cells that lack mitochondria or under anaerobic conditions. However, in the presence of mitochondria and oxygen, the NADH produced in glycolysis can participate in the shuttle pathway for the synthesis of ATP. If the cytosolic NADH uses Malate aspartate shuttle, 3 ADP are generated from each molecule of NADH. This is in contrast to glycerol phosphate shuttle that produces only 2 ADP. Irreversible steps in glycolysis. Irreversible steps in glycolysis. Most of the reactions in glycolysis are irreversible. However, the 3 steps catalyzed by the enzymes hexokinase or glucokinase, phosphofructokinase, and pyruvate kinase are irreversible. These 3 stages mainly regulate glycolysis. The reversal of glycolysis with alternate arrangements made of the 3 irreversible stages leads to the synthesis of glucose from pyruvate gluconeogenesis. Regulation of glycolysis. The 3 enzymes, namely hexokinase and glucokinase, phosphofructokinase, and pyruvate kinase, catalyzing the irreversible reactions, regulate glycolysis. Hexokinase is inhibited by glucose 6-phosphate. The enzyme prevents the accumulation of glucose 6-phosphate due to product inhibition. Glucokinase, which is specifically phosphorylates glucose, probably through the involvement insulin induces glucokinase. Phosphofructokinase, PFK, is the most important regulatory enzyme in glycolysis. This enzyme catalyzes the rate-limiting committed step. PFK is an allosteric enzyme regulated by allosteric effectors. ATP citrate and H+ ions, lower pH, are the most important allosteric inhibitors, whereas fructose 2,6-biphosphate, AMP, and pi are the allosteric activators. Role of fructose 2,6-biphosphate. Fructose 2,6-biphosphate, or F2,6-PP, is considered to be the most abundant regulatory factor activator for controlling PFK and ultimately glycolysis in the liver. F2,6-BP is synthesized from fructose 6-phosphate by the enzyme phosphofructokinase called PFK2. PFK1 is a glycolytic enzyme. F2,6-BP is catalyzed by fructose 2,6-biphosphatase. The function of synthesis and degradation of F2,6-BP is brought out by a single enzyme, same polypeptide, with 2 active sites, which is referred to as bifunctional enzyme. In fact, the combined name of phosphofructokinase 2, fructose 2,6-biphosphatase, is used to refer to the enzyme that synthesizes and degrades F2,6-BP. The activity of PFK2 and fructose 2,6-biphosphatase is controlled by covalent modification, which in turn is regulated by a cyclic AMP. CAMP is a second messenger for certain hormones. Cyclic AMP brings about dephosphorylation of the bifunctional enzymes, resulting in inactivation of active site responsible for the synthesis of F2,6-BP, but activation of the site responsible for the hydrolysis of F2,6-BP. This formula shows that CAMP in the presence of phosphofructokinase leads to phospho 2,6-biphosphate and phospho 6-phosphate. And finally, production of this. So it's a regulation of 2,6-biphosphate. Pyruvate kinase also regulates glycolysis. This enzyme is inhibited by ATP and activated by F1,6-BP. Pyruvate kinase is active A in dephosphorylated state and inactive B in phosphorylated state. Inactivation of pyruvate kinase by phosphorylation is brought about by CAMP-dependent protein kinase. The hormone glucagon inhibits hepatic glycolysis by this mechanism. Pasteur effect. The inhibition of glycolysis by oxygen, aerobic condition, is known as Pasteur effect. This effect was discovered by Louis Pasteur more than a century ago while studying fermentation by yeast. He observed that when the anaerobic yeast cultures metabolizing yeast were exposed to air, the utilization of glucose decreased by nearly 7-fold. In the aerobic condition, the levels of glycolytic intermediates from fructose 1,6-biphosphate onwards decrease while the earlier intermediates accumulate. This clearly indicates that Pasteur effect is due to the inhibition of the enzyme phosphofructokinase. The inhibitory effect of citrate and ATP produced in the presence of oxygen on phosphofructokinase explains the Pasteur effect. The Crabtree effect, the phenomenon of inhibition of oxygen consumption by the addition of glucose to tissues having high aerobic glycolysis, is known as Crabtree effect. Basically, this is opposite to that of Pasteur effect. Crabtree effect is due to increased competition of glycolysis for inorganic phosphate, PI, and NAD+, which limits the availability for phosphorylation and oxidation. Rappaport-Leubering Cycle. This is a supplementary pathway to glycolysis which is operative in the erythrocytes of man and other mammals. Rappaport-Leubering cycle is mainly concerned with the synthesis of 2,3-biphosphoglycerate 2,3-BPG in the RBC. So remember, Rappaport-Leubering cycle happens in the erythrocytes. So 1,3-biphosphoglycerate produced in glycolysis is converted to 2,3-BPG by the enzyme 2,3-biphosphoglycerate mutase. So 2,3-BPG is hydrolyzed to 3-phosphoglycerate by biphosphoglycerate phosphatase. Note, there's a difference between the usages biphosphate and diphosphate. A biphosphate has two phosphates held separately. 2,3-BPG in contrast to diphosphate, example ADP, where the phosphates are linked together. And 1,3-glucose, 1/2 glucose gives glyceraldehyde 3-phosphate. And it is now believed that biphosphoglycerate mutase is a bifunctional enzyme which mutates with main phosphatase activities catalyzed by two different sites present on the same enzyme. About 15 to 25% of the glucose converted to lactate in erythrocytes goes via 2,3-BPG synthesis. Significance in 2,3-BPG. Production of 2,3-BPG allows the glycolysis to proceed without the synthesis of ATP. This is advantageous to erythrocytes since glycolysis occurs when the need for ATP is minimal. Rappaport-Leubering cycle is therefore regarded as a shunt pathway of glycolysis to dissipate or waste the energy not needed by erythrocytes. 2,3-BPG, however, is not a waste molecule in RBC. It combines with hemoglobin Hb and reduces Hb affinity with oxygen. Therefore, in the presence of 2,3-BPG, oxyhemoglobin unloads more oxygen to the tissues. Increase in erythrocytes, 2,3-BPG is observed in hypoxic conditions, high altitude, fetal tissues, anemic conditions, etc. In all these cases, 2,3-BPG will enhance the supply of oxygen to the tissues. Glycolysis in the erythrocytes is linked with 2,3-BPG, production and oxygen transport. In the deficiency of the enzyme hexokinase, glucose is not phosphorylated. Hence, the synthesis and concentration of 2,3-BPG are low in RBC. The hemoglobin exhibits high oxygen affinity in hexokinase defective patients. Hexokinase defective patients. On the other hand, in the patients with pyruvate kinase deficiency, the level of 2,3-BPG in erythrocytes is high, resulting in low oxygen affinity. Conversion of pyruvate to acetyl-CoA. Pyruvate is converted to acetyl-CoA by oxidative decarboxylation. This is an irreversible reaction catalyzed by a multi-enzyme complex known as pyruvate dehydrogenase complex PDH, which is found only in the mitochondria. High concentrations of PDH are found in cardiac muscle and kidney. The enzyme PDH requires 5 cofactors coenzymes, namely TPP, lipoamide, FAD, coenzyme A, and NAD plus lipoamide contains lipoic acid linked to alpha amino group of lysine. The overall reaction of PDH is pyruvate plus NAD plus CoA. The presence of PDH gives acetyl-CoA plus CO2 plus NADH plus H+. Reactions of PDH complex. The sequence of reactions is brought about by different enzymes of RDH complexes in association with the coenzymes is depicted in the figure. Pyruvate is decarboxylated to give dihydrolipoyl transacetylase brings about the formation of acetyl-lipoamide from hydroxyethyl TPP and then catalyzes the transfer acetyl group to coenzyme A to produce acetyl-CoA. The cycle is complete when reduced lipoamide is converted to oxidized lipoamide, dihydrolipoyl dihydrogenase, transferring the reducing equivalents to FAD, FADH2. In turn, transfers the reducing equivalents to NAD plus to give NADH plus H plus, which can pass through the respiratory chain to give 3 ATP, 6 ATP from 2 moles of pyruvate formed from glucose by oxidative phosphorylation. The intermediates of PDH catalyzed reaction are not free, but bound to the enzyme complex. In mammals, the PDH complex has an approximate molecular weight of 9 × 10⁶. It contains 60 molecules of dihydrolipoyl transacetylase and about 20 to 30 molecules each of the Other two enzymes, pyruvate dehydrogenase and dihydrolipoyl dehydrogenase. A comparable enzyme with the PDH is aketoglutarate dehydrogenase, complex of citric acid cycle, which catalyzes the oxidative carboxylation of alpha-ketoglutarate to succinyl-CoA. Biochemical importance of PDH. Lack of TPP due to deficiency of thiamine inhibits PDH activity, resulting in the accumulation of pyruvate. In the thiamine-deficient alcoholics, pyruvate is rapidly converted to lactate, resulting in lactic acidosis. In patients with inherited deficiency of PDH, lactic acidosis, usually after glucose load is observed. The PDH activity can be inhibited by arsenic and mercuric ions. This is brought about by a binding of these ions with SH groups of lipoic acid. And pyruvate is a key metabolite. Besides its conversion to acyl-CoA utilized in a wide range of metabolic reactions, citric acid cycle, fatty acid synthesis, etc., pyruvate is a good substrate for gluconeogenesis. The next topic will be biological oxidation. The oxidation is defined as the loss of electrons and reduction as the gain of electrons. This may be illustrated by the interconversion of ferrous ion Fe2+ to ferric ion Fe3+. Fe2+ oxidation or reduction. When oxidation an electron is lost but accepted reduced, where other ways in reduction it is accepted Fe3+. The electron lost in the oxidation is accepted by an acceptor, which is said to be reduced. Thus, the oxidation reduction is a tightly coupled process. The general principle of oxidation reduction is applicable to biological systems also. The oxidation of NADH to NAD+ coupled with the reduction of FMN to FMNH2 is illustrated here. That's shown in the diagram. NADH plus H+ gives NAD, the present releasing hydrogen and 2 electrons. FMN is converted to FMNH2. In the above illustration, there are two redox pairs, NADH and NAD+, and FMN and FMNH2. The redox pairs differ in their tendency to lose or gain electrons. Redox potential E0. The oxidation reduction potential, or simply redox potential, is a quantitative measure of the tendency of a redox pair to lose or gain electrons. The redox pairs are assigned a specific standard redox potential EO volts at pH 7 and 25 degrees centigrade. The redox potential of some biologically important redox systems are given in Table 3, as seen here. The more negative redox potential represents a greater tendency of reductant to lose electrons. On the other hand, a more positive redox potential indicates a greater tendency of oxidant to accept electrons. The electrons flow from a redox pair with more negative EO to another redox pair with more positive EO. The redox potential E is directly related to the change in the free energy delta G0. So standard redox potential of various oxidation reduction systems. Right? Electron transport chain. The energy-rich carbohydrates, particularly glucose, fatty acids, and amino acids, undergo a series of metabolic reactions and finally get oxidized to covalent CO2 and H2. The reducing equivalents from various metabolic intermediates are transferred to coenzymes NAD+ and FAD to produce, respectively, NADH and FADH+. The latter two reduced coenzymes pass through the electron transport chain EDC of respiratory chain and finally reduce oxygen to water. The passage of electrons through the ATC is associated with the loss of free energy. A part of this free energy is utilized to generate ATP from ADP and PI. Mitochondria, the powerhouse of the cell. The mitochondria are the centers of metabolic oxidative reactions to generate reduced coenzymes. NADH and FADH2, which are in turn utilized in EDC to liberate energy in the form of ATP. For this reason, mitochondria is appropriately regarded as the powerhouse of the cell. Mitochondrial organization. The mitochondrion consists of 5 distinct parts. These are the outer membrane, the inner membrane, the intermembrane space, the cristae, and the matrix. The electron transport chain and the ADP synthetic system are located And the inner mitochondrial membrane, which is a specialized structure rich in proteins. It is impermeable to ions, H plus, potassium and sodium, and small molecules ADP and ATP. The membrane is highly folded to form cristae. The surface area of inner mitochondrial membrane is greatly increased due to cristae. The inner surface of the inner mitochondrial membrane possesses specialized particles. They look like lollipops. The phosphorylating subunits, which are the centers for ATP production. The mitochondrial matrix, the inner ground substance, forms the matrix of the mitochondria. It is rich in the enzymes responsible for the citric acid cycle, beta oxidation of fatty acids and oxidation of amino acids. Structural organization of respiratory chain. The inner mitochondrial membrane can be disrupted into 5 distinct respiratory or enzyme complexes denoted as complex 1, 2, 3, 4, and 5. The complexes 1 and 4 are carriers of electrons, while complex 5 is responsible for ATP synthesis. Besides these enzyme complexes, there are certain mobile electron carriers in the respiratory chain. These include NADH, coenzyme Q, cytochrome C, and oxygen. The enzyme complexes 1, 4, and the mobile carriers are collectively involved in the transport of electrons, which ultimately combine with the oxygen to produce water. The largest proportion of the oxygen supplied to the body is utilized by the mitochondria for the operation of electron transport chain. Nicotinamide nucleotides of the 2 coenzymes NAD plus and NAD plus derived from the vitamin niacin. NAD plus is more actively involved in the ETC. NAD plus is reduced to NADH plus H plus by dehydrogenases with the removal of 2 hydrogen atoms from the substrate AH2. Substrates include glyceraldehyde, 3-phosphate, pyruvate, isocitrate, alpha-ketoglutarate, and malate. AH2 plus NAD plus gives A plus NADH and H plus. NADPH plus H produced by NADP-dependent dehydrogenase is not usually a substrate for ETC. NADPH is more effectively utilized for anabolic reactions, example fatty acid synthesis and cholesterol synthesis. Flavor proteins, the enzyme NADH dehydrogenase is a flavor protein with FMN as the prostatic group. The coenzyme FMN accepts 2 electrons and a proton to form FMN H2. NADH dehydrogenase is a complex enzyme closely associated with non-heme iron proteins, NHI, or iron sulfur proteins. FES, NADH plus H plus FMN gives 7 NAD plus FMNH2. Succinate dehydrogenase, succinate coenzyme Q reductase, is an enzyme found in the inner mitochondrial membrane. It is also a flavor protein with FAD as the coenzyme. This connects up 2 hydrogen atoms, 2H plus 2E from succinate plus FAD gives fumarate and FADH2. Iron sulfur proteins. The iron sulfur FeS proteins exist in the oxidized Fe3 plus or reduced Fe2 plus state. About half a dozen FeS proteins, that is iron sulfide proteins, connected with the respiratory chain have been identified. However, the mechanism of action of iron sulfur proteins in the ETC is not clearly understood. One FeS participates in the transfer of electrons from FMN to coenzyme Q. Other FeS proteins associated with the cytochrome B and cytochrome C participate in the transfer of electrons. Coenzyme Q. Coenzyme Q is also known as ubiquinone since it is ubiquitous in living systems. It is a quinone derivative with a variable isoprenoid side. The mammalian tissues possess a quinone with 10 isoprenoid units, which is known as coenzyme Q10, CoQ10. Coenzyme Q is a lipophilic electron carrier. It is an accepted electron from the FMNH2 produced the ETC by NADH dehydrogenase or FADH2 produced outside ETC. Example succinate dehydrogenase, acyl-CoA dehydrogenase. Coenzyme Q is not found in the mycobacteria. Vitamin K performs similar functions as the coenzyme Q in these organisms. Coenzyme Q has no vitamin precursor in animals. It is directly synthesized in the body. Cytochromes. The cytochromes are conjugated proteins containing heme group. The latter consists of porphyria ring with the iron atom. The heme group of cytochromes differ from those found in the structure of hemoglobin and myoglobin. The iron of heme in cytochromes is also natively oxidized Fe3 plus and reduced Fe2 plus, which is essential for the transport of electrons In the ETC, this is in contrast to the heme ion of hemoglobin and myoglobin, which remains in the ferrous Fe2 state. 3 cytochromes were initially discovered from the mammalian mitochondria. They were designated as cytochrome A, band C, depending on the type of heme present in the respective absorption spectrum. Additional cytochromes such as C1, B1, B2, A3, etc. were discovered later. The electrons are transported from coenzyme Q to cytochromes in the order B, C1, C, A, and A3. The property of reversible oxidation reductions of heme ion Fe2+, Fe3+ present in cytochromes allows them to function as effective carriers of electrons in He. Cytochrome C, molecular weight 13,000, is a small protein containing 104 amino acids and a heme group. It is a central member of ETC with an intermediate redox potential. It is rather loosely bound to inner mitochondrial membrane and can be easily extracted. Cytochrome A and A3. The term cytochrome oxidation is frequently used to collectively represent cytochrome A and A3, which is the terminal component of He. Cytochrome oxidase is the only electron carrier, the heme ion of which can directly react with molecular oxygen. Besides heme with iron, the oxidase also contains copper that undergoes oxidation reduction Cu2+. During the transport of electrons, in the final stage of He, the transport of electrons, the free protons and the molecular oxygen combine to produce water. Cytochrome A and A1. The term cytochrome A and A3, this term cytochrome oxidase is frequently used to collectively represent cytochrome A and A3, which is the terminal component of He. Cytochrome oxidase is the only electron carrier, the heme ion of which can directly react with molecular oxygen, besides heme with iron. This oxidase Besides heme with iron, this oxidase also contains copper that undergoes oxidation reduction Cu2 to Cu. During the transport of electrons in the final stage of He, the transport of electrons, the free protons, and the molecular oxygen combine to produce water. The next topic for today is oxidative phosphorylation. The transport of electrons through the ETC is linked with the release of free energy. The process of synthesizing ATP from ADP and PI, it's organic phosphorus, coupled with the electron transport chain is known as oxidative phosphorylation. The complex V of the inner mitochondrial membrane is a site of the oxidative phosphorylation, PO. The rare P-is-to-O ratio phosphor is to oxygen ratio refers to the number of inorganic phosphate molecules utilized for ATP. For ATP generation, for every atom of oxygen consumed, more appropriately, P-is-to-O ratio represents the number of molecules of ATP synthesized per pair of electrons carried through ETC. The mitochondrial oxidation of NADH with a P-is-to-O ratio of 3 can be represented by the following equation. NADH plus H plus half O2 plus 3 ADP plus 3 organic phosphate gives NAD plus 3 ADP plus 4 H2. O. P-is-to-O ratio of 2 is assigned to the oxidation of FADH2. Note, although yet to be approved beyond doubt, some workers suggest P-is-to ratio of 2.5 for NADH plus H plus and 1.5 for FADH2 based on the proton translocation. Sites of oxidative phosphorylation in ETC. There are three reactions in the He that are exergonic to result in the synthesis of 3 ATP molecules. The three sites of ATP formation in ETC are oxidation of FMNH2 by coenzyme Q, oxidation of cytochrome B by cytochrome C1, and cytochrome oxidase reaction. Each one of the above reactions represents a coupling site for ATP production. There are only two coupling sites for the reaction mentioned now of oxidation of FADH2. P is to O ratio, since the first site is bypassed. Energetics of oxidative phosphorylation. The transport of electrons from redox pair NAD plus bar NADHE E0 is equal to 0.32 to finally the redox pair of half O2 H2O E0 equals 0.82. May be simplified and represented in the following equation. Half oxygen plus NADH plus H plus gives water plus NAD plus. The redox potential difference between these two redox pairs is 1.14 V, which is equivalent to an energy 52 cals per mole. 3 ATP are synthesized in the ETC when NADH is oxidized, which equals to 21.9 cals. Each ADP is equal to 7.3 cals. The efficiency of energy conservation is calculated as 21.9 into 100 per 52, which is equal to 42%. Therefore, when NADH is oxidized, about 42% of energy is trapped in the form of 3 ADP, and the remaining is lost as heat. Mechanism of oxidative phosphorylation. Several hypotheses have been put forth to explain the process of oxidative phosphorylation. The most important among them, namely chemical coupling, conformational coupling, and chemi-osmotic, are discussed below. Chemical coupling hypothesis. This hypothesis was put forth by Edward Slater in 1953. According to chemical coupling hypothesis, during the course of electron transfer and respiratory chain, a series of phosphorylated high-energy intermediates are first produced, which are utilized for the synthesis of ATP. These reactions are believed to be analogous to the substrate-level phosphorylation that occurs in glycolysis or citric acid cycle. However, this hypothesis lacks experimental evidence, since all attempts so far to isolate any one of high-energy intermediates have not been successful. Conformational coupling hypothesis. Paul Boyer proposed this hypothesis in 1964. As a consequence of electron transfer, conformational change in the membrane protein is induced. This is believed to be responsible for synthesis of ATP. The protein attains a high energy conformation due to electron transfer, which when reverted back to a random state, low energy, results in the production of ATP from ADP plus Pi, shown in the figure. There is some evidence demonstrating conformational changes in mitochondrial membrane proteins. However, there is no conclusive proof that such changes may be actually responsible for ATP synthesis. Chemi-osmotic hypothesis. This mechanism, originally proposed by Peter Mitchell, 1961, is now widely accepted. It explains how the transport of electrons through the respiratory chain is effectively utilized to produce ATP from ADP and Pi, that is phosphonic organic phosphate. The concept of chemiosmotic hypothesis is comparable with the energy stored in a battery separated by positive and negative charges. Proton gradient. The inner mitochondrial membrane as such is impermeable to protons H plus and hydroxyl ions OH plus negative. The transport of electrons through ADC is coupled with the translocation of protons H plus across the inner mitochondrial membrane, coupling membrane. For the matrix to the intermembrane space, the pumping of protons results in an electrochemical or proton gradient. This is due to the accumulation of more H plus ions, low pH, on the outer side of the inner mitochondrial membrane than the inner side. The proton gradient developed due to the electron flow in the respiratory chain is sufficient to result in the synthesis of ATP from ADP and Pi. Oxidation and reduction, O by R loops or redox loops. It is postulated that the ETC is folded into 3 oxidation reduction loops corresponding to complex 1, 3, and 4. Each loop is a coupling site and consists of a hydrogen carrier and an electron carrier. 6 protons are translocated for each electron pair transfer from NADH plus H plus to oxygen to 3 oxidation reduction loops along with the carriers of protons and electrons are depicted in the figure. That figure would be figure 11, which comes down, we'll show you later. It's figure 9, okay? The first pair of protons, 2H plus, are translocated via FMN, and the second NH, 2H plus via coenzyme. This is the figure. ETC with redox loops, tentative in chemiosmotic hypothesis. Okay, take a screenshot and study on it. There is still some controversy regarding the carrier of the third proton pair designated as X in the figure 11. Some workers believe that the coenzyme Q is involved here. Coenzyme Q is a lipid-soluble small molecule and can freely move across the inner mitochondrial membrane. A cyclic process referred to as coenzyme Q cycle is said to be operative for translocation of protons at two oxidation reduction loops. Enzyme system for ATP synthase. For ATP synthesis. ATP synthase present in the complex 5 utilizes the proton gradient for the synthesis of ATP. This enzyme is also known as ATPase since it can hydrolyze ATP to ADP and pi. That's inorganic phosphate. Organic phosphate. ADP synthase is a complex enzyme and consists of two functional subunits, namely F1 and F0. Its structure is comparable with the lollipops. We mentioned this before, so it's good for a recap. The protons that accumulate on the intermembrane space re-enter the mitochondrial matrix, leading to the synthesis of ATP. This process, involving ATP synthase, occurs in 3 stages. Subunit F0 of ATP synthase translocates protons re-entry into the matrix. F1 subunit catalyzes the formation of ATP from ADP and pi. This is believed to involve a conformational change in F0 and F1. Go to the next page. F0 and F1 subunits coordinate with each other and couple the disappearance of proton gradients with ATP synthase. That's a diagram for that. Evidence in support of chemiosmotic hypothesis. Many experimental observations support the chemiosmotic hypothesis. The intact inner mitochondrial membrane is responsible for oxidative phosphorylation. The inner mitochondrial membrane is impermeable to protons, hydrogen, and other ions, OH, Ka, Cl, etc. The ATP can be generated by increasing the H ion concentration by adding acid on the outside. On the outside of mitochondria. Let me repeat that for you. ATP can be generated by increasing the hydrogen ion concentration by adding acid on the outside of mitochondria. Certain compounds which increase the permeability of inner mitochondrial membrane to protons inhibit the ATP synthesis. For example, 2,4-dinitrophenol. Inherited disorders of oxidative phosphorylation. It is estimated that about 100 polypeptides are required for oxidative phosphorylation. Of these, 13 are coded by mitochondrial DNA, mtDNA, and synthesized in the mitochondria, while the rest are produced in the cytosol, coded by nuclear DNA and transported. Mitochondrial DNA is maternally inherited since mitochondria from the sperm do not enter the fertilized ovum. Mitochondrial DNA is about 10 times more susceptible to mutations than nuclear DNA. Mitochondrial DNA mutations are more commonly seen in tissues with higher rate of oxidative phosphorylation. That is, example, central nervous system, skeletal, heart, liver, heart muscles, and liver. Liver's hereditary optic neuropathy is an example for mutations in mitochondrial DNA. This disorder is characterized by loss of bilateral vision due to neuroretinal degeneration. Inhibitors of electron transport chain. Many site-specific inhibitors of ATG have contributed to the present knowledge of mitochondrial respiration. Selected examples of these inhibitions are given in figure 7. The inhibitors bind to one of the components of ETC and block the transport of electron. This causes the accumulation of reduced components before the inhibitor and blockade step, the inhibitor blockade step, and oxidized dark components after that step. The synthesis of ATP phosphorylation is dependent on electron transport. Hence, all the site-specific inhibitors of ETC also exhibit ATP function formation. 3 possible sites of action of the inhibitors of ETC are identified. They are 1. NADH and coenzyme Q, fish poison rotenone, barbiturate drug emityl, and antibiotic pyridine A inhibit at this site. Between cytochrome B and C, antimycin A, an antibiotic, British anti-leukocyte, BAL, an antidote used in or against Wargash are the 2 important inhibitors of the site between cytochrome B and C1. Inhibitors of cytochrome oxidase, cytochrome carbon monoxide cyanide, hydrogen sulfide, and acide effectively inhibit cytochrome oxidase. Carbon monoxide reacts with the reduced form of the cytochrome, while cyanide and acide react with oxidized form. Inhibitors of oxidative phosphorylation, uncouplers. The mitochondrial transport of electrons is tightly coupled with oxidative phosphorylation ATP synthase. In other words, oxidation and phosphorylation proceed simultaneously. There are certain compounds that can uncouple or delink the electron transport from oxidative phosphorylation. Such compounds, known as uncouplers, increase the permeability of inner mitochondrial membrane to protons, H+. Thus, an uncoupler allows electron transport but blocks the establishment of proton gradient across the inner mitochondrial membrane. The result is that ATP synthesis does not occur. The energy link with the transport of electrons is dissipated as heat. The uncouplers allow, often at accelerated rate, oxidation of substrates via NADH or FADH2 without ATP formation. The uncoupler 2,4-dinitrophenol DNP has been extensively studied. It is a small lipophilic molecule. DNP is a proton carrier and can effectively diffuse through the inner mitochondrial membrane. The other uncouplers include dinitrocresol, pentachlorophenol, trifluorocarbonyl cyanide, and phenylhydrazone, FCCP. Trifluorocarbonyl cyanide phenylhydrazone as FCCP. The last compound, FCCP, is said to be 100 times more effective as an uncoupler than dinitrophenol. When administered in high doses, the drug aspirin acts as an uncoupler. So the next one is physiological uncouplers. Certain physiological substances which act as uncouplers at high concentration have been identified. These include the hormone thyroxine and long-chain free fatty acids. The unconjugated bilirubin is also believed to act as an uncoupler. This is, however, yet to be proved beyond doubt. Significance of uncoupling. Uncoupling of respiration from oxidative phosphorylation under natural conditions assumes biological significance. The maintenance of body temperature is particularly important in hairless animals, hibernating animals, and the animals adapted to cold. These animals possess a specialized tissue called brown adipose tissue in the upper back and neck portions. The mitochondria of brown adipose tissue are rich in electron carriers and are specialized to carry out an oxidation, which is uncoupled from phosphorylation. This causes liberation of heat, where fat is oxidized in the brown adipose tissue. The presence of active brown adipose tissue in certain individuals is believed to protect them from becoming obese. The excess calories consumed by these people are burnt and liberated as heat instead of being stored as fat. Ionophores. The term ionophores is used collectively to represent the lipophilic substances that promote the transport of ions across biological membranes. All the uncouplers described above are in fact proton ionophores. The antibiotics valinomycin and nigersin act as ionophores for K plus ions. Both of these compounds are also capable of dissipating proton gradient across the inner mitochondrial membrane and inhibit oxidative phosphorylation. Other inhibitors of oxidative phosphorylation, oligomycin, this antibiotic prevents the mitochondrial oxidation as well as phosphorylation. It binds with the enzyme ATP synthase or synthetase and blocks the proton H plus channels. It thus prevents translocation re-entry of protons into mitochondrial matrix. Due to this, protons get accumulated at higher concentration in the intermembrane space. Electron transport, respiration ultimately stops since proton cannot be pumped out against deep proton gradients. The action of oligomycin indicates that oxygen and phosphorylation are tightly coupled processes. Atractyloside. This is a plant toxin and inhibits oxidative phosphorylation by an indirect mechanism. The presence of adequate concentration ADP and PI in the mitochondrial matrix and the transport of ATP into the extramitochondrial system are needed for phosphorylation to proceed continuously. Adenine dinucleotide carrier system facilitates the transfer of ATP and ADP. Atractyloside inhibits adenine dinucleotide carrier and thus blocks the adequate supply of ADP, thereby preventing phosphorylation. Transport of reducing equivalence shuttle pathways. The inner mitochondrial membrane is impermeable to NADH. Therefore, the NADH produced in the cytosol cannot directly enter the mitochondria. Two pathways, namely glycerol phosphate shuttle and malate aspartate shuttle, are operated to do this job. They transport the reducing equivalents from cytosol to mitochondria, not vice versa. The shuttle pathways are not operated to the same extent in all the tissues. The malate aspartate shuttle predominantly occurs in liver and heart, whereas the glycerol phosphate shuttle operates in muscle and brain. The glycerol phosphate shuttle. Cytosolic glycerol 3-phosphate dihydrogenase oxidizes NADH to NAD+. The reducing equivalents are transported through glycerol 3-phosphate into the mitochondria. Glycerol 3-phosphate dehydrogenase present on outer surface of inner mitochondrial membrane reduces FAD to FADH. Dihydroxyacetone phosphate escapes into the cytosol and the shuttling continues as depicted in the figure, gets oxidized via EDC to generate 2-ADP. Malate aspartate shuttle. In the cytosol, oxaloacetate accepts the reducing equivalents NADH and becomes malate. Malate then enters the mitochondria where it is oxidized by mitochondrial malate dehydrogenase. In this reaction, NADH and oxaloacetate are regenerated. NADH gets oxidized via electron transport chain and 3-ATP are produced. This is in contrast to glycerol phosphate shuttle where only 2-ADP are produced in the mitochondria. Oxaloacetate participates in transamination reactions with glutamate to produce aspartate and alpha-ketoglutarate aspartate. It enters the cytosol and transaminates with alpha-ketoglutarate to give oxaloacetate and glutamate. The malate aspartate shuttle is shown here, as seen here, right? Now we're going on to the next topic of coupled reactions and high energy compounds, which are energy-rich compounds, right? Metabolism is essentially a linked series of chemical reactions that begins with a particular molecule and converts it into some other molecule or molecules in a carefully defined fashion. There are many such defined pathways in the cell, and we will examine a few of them in detail here. These pathways are interdependent, and the activity is coordinated by exquisitely sensitive means of communication in which allosteric enzymes are predominant. We can divide metabolic pathways into two broad classes, those that convert energy into biologically useful forms and those that require inputs of energy to proceed. Although this division is often imprecise, it is nonetheless a useful distinction in an examination of metabolism. Those reactions that transform fuels into cellular energy are called catabolic reactions, or more generally catabolism. fuel, carbohydrates, fats, example, undergo catabolism to give carbon dioxide, water, and useful energy. Those reactions that require energy, such as the synthesis of glucose, fats, or DNA, are called anabolic. Reactions of anabolism, the useful forms of energy that are produced in catabolism are employed in anabolism to generate complex structures from simple ones or energy-rich states from energy-poor ones. Useful energy plus small molecules, anabolism, complex molecules. Some pathways can be either anabolic or catabolic. Depending on the energy conditions in the cell, they are referred to as amphibolic pathways. Thermodynamically unfavorable reaction can be driven by a favorable reaction. How are specific pathways constructed from individual reactions? A pathway must satisfy minimally two criteria. 1. The individual reactions must be specific. And 2. The entire set of reactions that constitute the pathway must be thermodynamically favored. A reaction that is specific will yield only one particular product or set of products from its reactants. The function of enzymes is to provide this specificity. The thermodynamics of metabolism is most readily approached in terms of free energy. A reaction can occur spontaneously only if delta G, the change in free energy, is negative. Recall that delta G for the formation of products C and D from substrates A and B is given here by delta G equal delta G0 prime plus RT basic logarithm C into D by A into B. Thus, the delta G of reactions and products or the nature of the reactions and products expressed by the delta G0 term, the standard free energy change on their concentrations expressed by the second term. An important thermodynamic fact is that the overall free energy change for a chemically coupled series of reactions is equal to the sum of the free energy changes of the individual steps. Consider the following reactions for that. A reversible B plus C gives delta G0 prime is equal to 5 kilocalories. And B reversible D delta G0 prime is minus 8 kilocalories. And the difference is A differential reversible C plus D delta G0 prime gives minus 3 kilocalories. Right? Using standard conditions, A cannot be spontaneously converted into B and C because DG is positive. However, the conversion of B into D under standard conditions is thermodynamically feasible because free energy changes are additive. The conversion of A into C and D has a delta G0 of 3 kilocalories per mole, which means that it can occur spontaneously under standard conditions. Thus, a thermodynamically unfavorable reaction can be driven by a thermodynamically favorable reaction to which it is coupled. In this example, the chemical intermediate R, B, common to both reactions couples the reactions. Thus, metabolic pathways are formed by the coupling of enzymes, catalyst reactions, such that the overall free energy of the pathway is negative. ATP is the universal the currency of free energy in biological systems. Just as commerce is facilitated by the use of a common currency, the commerce of the cell metabolism is facilitated by the use of common energy currency, adenosine triphosphate ATP. Part of the free energy derived from the oxidation of foodstuffs and from light is transformed into a highly accessible molecule, which acts as the free energy donor in most energy requiring processes such as motion, active transport, or biosynthesis. ATP is a nucleotide consisting of an adenine, a ribose, and a triphosphate unit. The active form of ATP is usually a complex of ATP with Mg2 or manganese. In considering the role of ATP as an energy carrier, we can focus on its triphosphate moiety. ATP is the energy-rich molecule because its triphosphate unit contains 2 phosphoanodride bonds. A large amount of free energy is liberated when ATP is hydrolyzed to adenosine diphosphate ADP and orthophosphate P1, or when ATP is hydrolyzed to adenosine monophosphate AMP and pyrophosphate PP1. So ATP plus water gives AMP plus PP1, as orthophosphate. Free energy is equal to minus 10.9 kilocalories, mole raised to minus 1, and ADP plus that water gives ADP plus P1, delta G0 equal to minus 7.3 kilocalories. The precise DG for these reactions depends on the ionic strength of the medium and on the concentrations of magnesium and other metal ions under typical cellular concentrations. The actual DG for these hydrolyses is approximately minus 12 kilocalories. The free energy liberated in the hydrolysis of ATP is harnessed to drive reactions that require an input of free energy, such as muscle contraction. In turn, ATP is